नमस्कार, मैं डॉक्टर अवधेश कुमार श्रीवास्तव, असिस्टेंट प्रोफेसर, डिपार्टमेंट ऑफ बॉटनी एंड माइक्रोबायोलॉजी, गवर्नमेंट DTPG College, Uttar Pradesh. Today we have gathered to discuss a new topic regarding practical to determine diversity indices by Sanam-Vanner methods. And after we will discuss about the concentration of dominance, species richness, and equability and beta diversity. So let us talk together. If we talk about alpha diversity, so in alpha diversity, it is the diversity of each site. It is local species of any pool. Suppose if you are wandering in your garden, that is known as alpha diversity. If you move in a farmhouse, so whatever the local species pool is there, they are known as alpha diversity. As per the beta diversity is concerned, it is the difference between species composition among the sites. Let us discuss. There is a landscape. The total, there are river, there is a forest, there is a mountain, everything, whatever you are visualizing. So that is the gamma one. So if you divide gamma and alpha diversity, it becomes beta. So it is the difference in species composition. It is the difference in the species composition among the sites. Suppose that this is alpha 1 and whatever the differences is there in between these two. This is alpha, this is also alpha. Suppose this is a forest, this is a grassland. So whatever you are studying is alpha diversity, and the difference among these two is known as beta. And if we discuss all the other alpha together, then it is known as gamma diversity. This is the entire landscape. So alpha and gamma diversity are straightforward. If you see a local area, then it is alpha. If you see a broad area, that is gamma. And whatever differences is there, that is known as beta diversity. Let us see in this diagram. See, this is the alpha diversity of a community, because here we are taking this small patch. This is also a type of alpha diversity. But the difference between these two, what are the commonality and what are the difference? So this is the beta diversity between the community. And this one is the alpha diversity of a community. So this is also alpha. This is also alpha. This is also alpha. This is also alpha. So all are alpha. So whatever the difference in between these two is known as beta. And if you take the total area, I mean, if you change this color, if you are taking this entire landscape, then it becomes gamma diversity. See, if you think about these three areas, this is one, this is another, this is another. So how many structures are there? So it is consist of one type. This is another two. This one is the three. This one is the 4, and this one is the 5. So suppose if there are 5 type of trees, so 5 different type of things are there. So community A is made up of 5 different cells. This is community B. And you can also visualize these are the different structures, and they are also of 5 types. So if we talk about beta diversity, 5 minus 5 is equal to 0. But if we talk about this community, this is the community C, and it has only 3 structures. How there are 3 structures? This is one type, this is another type, this one is the another type. In this way, we have 3 different sets. So the alpha diversity is 3. So this is 3 different sets. So these are the alpha diversity. Now, see, like this, we are plotting this one alpha, another alpha, another alpha. We have three alpha sites. So what is the difference in between them is the beta one. And if you take total, then it becomes gamma. And if you're taking the total structure, then it becomes gamma. Whole data set is the gamma. So now I think it is very much clear to all of you. So in alpha, number of individuals will be there. If you take these as animal, these are as a plant. So likewise, how many type of alpha diversity is there? One type, and this is two type. One type, two type, three type. One type, two type, three type, four type, like this. Okay. So think about this. How many individuals are, how many types of individuals are here? Just think. So this becomes one, this becomes two, this is common one, and this is the three. So it is three. Think about this. So it is one, two, three, and four. So it is full. How many types of here? You don't count that number, otherwise it becomes four. So this one, this is the same copy, and this is the two. That's why it becomes two. So among this and this, this and this, this and this, so gamma is total 6 structure. See, one is red triangle, second is green rectangle, blue circle. So it becomes total, if one star is there, green circle is there, in this way, the gamma becomes 6. So if you think about this, the 6 by, so it becomes 3. How it is 3? Because this is one type, this is another type, and 3 structures, because you cannot see this in this area. Similarly, a star is also not present here, and yellow square is also not present here. So differences is 3. So this is beta diversity. Similarly here, this is present, this is not present. So in this case, beta is 4, but here it is 5. How it is 5? These 3 structures are not present here, and these 2 structures are also not present. So it becomes beta is equal to 5. So beta is the difference between 2 communities. Okay. Similarly here, this is site A, site B, site C, site D, and this forms a common area, this forms a common region. So the total 6 structures are there. So diversity is 6, alpha diversity. And in B area, same 6, but entire structure is 6, so it is 6, 6. So here, this beta is equal to 6 by 6, so it becomes 1 and 1. And similarly, if you think about this region CD, so alpha is 4. How it is 4? This is one type, this is another type, and this 3 and 4. So it becomes 4. And here, one type, two type, three type, and 4. So inside D, it is alpha is 3.8, like this. So if numbering is different, it might be possible. So in spite of 4, we are just counting, it may be less. So 6 by 4 is equal to 1.5, and 6 by 3.8 is 1.6. This is almost 4, but if you perform this experiment in a loose way, the result may comes like this. Okay. So if you think about biodiversity, I think it is very easy to isolate the alpha, beta, gamma diversity among 2 or 3 or any area. Okay. Have you seen this calculator? This is scientific calculator, and this button is very important for us because we are going to determine biodiversity by Shannon-Wenger method. So if you have this calculator, by using this button, you can get this result very easily. How? Suppose if I put quadrate in a field and number of plants are there, For example, I have taken three categories. This is one, this is another, this is one. Cyanodendrocalone, and the total number is 23. In Phyllanthus neuri, the total number is 27. And if it is Cytocordifolia, the total number is 12. Likewise, you may put so many names which is present as a biodiversity in that field. So if you add them, what will happen? This 3, 7, 10 plus 2, 12, carry over 1. So 2 plus 2, 4, 5, carry over 1, 6. So this is the number. This is the total number of individual present in that particular type of the alpha diversity or one particular. Because we are going to determine Shannon-Weiner diversity, which is represented by H bar or H. So how you can get it? This 23 is divided by 62. This 27 is divided by 62. From where this total number is 62. So if you are getting the percentage value of this one, if you are going to know the percentage value of this one, so by using a simple calculator, you can get a very percent in decimal. So percent in decimal is because we are not taking 100%, we have omitted this one. So it becomes 23 by 62. So it becomes 0.371. 27 divided by 62 is 0.435. And this 12 is divided by 62, it becomes 0.194. So this is the value which is known as PI. This is the value which is known as PI. And when you are going to get the lnx, that is natural logarithm value of this one, this one, this one, by using that calculator, you just put this button on and ln will come, and you just put the number by using this keyboard and by using these keyboards. You can very easily use these keyboards for the number. So what will happen? If you're putting ln x 0.371, ultimately it becomes minus 0.992. If you put ln of 0.435, it comes at 0.832. If you put 0.194, it becomes 1.640. Likewise. So if you add these figures, so what will happen? Now we are going to multiply this, that is PI, with lnx. With lnx, x is the value, that value. So if you multiply this with this, it becomes this 0.435, and whenever it is multiplied by minus 832, it becomes this. Similarly, when this is multiplied by this, it becomes 0.318. Now, summation of this PIX lnx x, whenever it is summed, in that condition, you will get the final product. So whenever this is summed, the sum value is like this. And the total figure comes in minus. So you just multiply them by minus. So it becomes plus. And this is the value of Shannon-Weiner, that is H. So very easily you can calculate Shannon-Weiner value of any type of diversity, that is for particular, alpha diversity. Okay, any problem? So this value is 1.048, that is slightly more than 1, And it is seen that whenever you're talking about diversity, if it is less than 1.5, up to 1.5 or less than 1.5, they are regarded as they are very low in their diversity. Of course, only 3 plants were taken into consideration, so they have low diversity. But when the result comes as 1.5 to 2.5, they are considered as, yes, moderate diversity. But whenever this value is more than 2.5, it is considered as high biodiversity, high diversity, high diversity. In our result, the result is 1 point, just we have noted, 1.048. So we have this result, that is 1.048. So it becomes less biodiversity is there. And this is the value of Shanor-Werner constant or indices, whatever you see. We have to find it only this peak. So this beaker is very easily calculated by the help of this scientific calculator. Within a moment, you are getting the result. Okay. Now, there are two important questions which is also associated with this, and they are, first is species composition. What is species composition? So different types of species present in a particular community. As for example, in our this result, how many plants was there? 1, 2, and 3. So what should be the community composition? 3. Now question comes, concentration of the dominance. Concentration of the dominance. So at a particular time, if a population which is highest in its density, is called dominance, and its density is called concentration of dominance. In our this example, we have taken this number and this pi as the concentration. So among these three plants, which each considered for highest value, naturally this one. So this value 0.435 has the maximum value. So according to our this problem, the Phyllanthus neruri has 0.435 value, dominance value. So it is considered as concentration of dominance was highest in Phyllanthus neruri. Okay. Some associated questions also comes for you, like species richness, which is represented by S. So species richness, and it is the value, number of species present in the community. So how many number of the species was present in this community? Because that is a community where we have put the quadrate and we have studied the number. So only 3 is the number. If the plants was 4, 5, 6 like that. So number may be changed. But in this experiment, the value of the species richness is only 3. Now question comes, species evenness, that is also known as equability, that is represented by E. So relative abundance of the species in that species that are present. It is also represented by this formula. So H, what is H? H is that value which we have just received here. This is the value of H. This is the value of H or H bar. That means this is the value of Shannon-Werner's diversity. So 1.048 was the value. And when it is subtracted from INSS, that is that particular species. S is that particular species. So if we talk about any one plant in our previous example, like this. So this 3, if you talk about this, so whenever you are subtracting 1.048 minus 0.368, this will be the result of what? Species evenness. 1.048 minus INS. INS is the value of all these INS. These are the INS 1, 2, 3. So you can put here and you can get the result. So this is about the species evenness. And what is species diversity? Here we have to study both the things, richness as well as evenness or equability, then they becomes species diversity. For you might have seen this in your laboratory. This is quadrate. The length and breadth are very common to each other. That's why the quadrate, and it is redivided into several groups. You can also make such type of quadrate by putting nail in field. So by this quadrate, these are the different formulas for percentage frequency, for relative frequency, relative density, abundance and relative abundance. And all the relative abundance, frequency and density are collectively known as IVI, important value index. If you want to isolate the relative abundance, your serial number 1, 2, 3, 4, 5, 6, you can put name of the plant on this axis, you can write. And by putting the quadrate, first quadrate, second quadrate, third quadrate, fourth quadrate, fifth quadrate, how many, suppose in first quadrate, the number is 2. In second, that was 0. In third, it is 3. Here it is 1. And in last, it is 5. So total number of individual of the species is 2, 3, 5, 6, and 11. So it becomes 11. Number of quadrate in which species was occurred, so it is only 4. So 11 by number of individual of a species, so it is 11. Number of in quadrate where it was present is divided by 4, and this is the value of the relative abundance. So it becomes 2 point something. So you can get the result by putting this method. So this is all about your practical, because in MSc practical, you might have seen the factors in your syllabus. The total topic was divided into these 4 or 5 important parts. The first was Shannon-Weiner concentration, and I think it is clear to all of you, isn't it? Shannon-Weiner methods, biodiversity. Concentration of dominance is also clear. Species richness is also clear. And equability and beta diversity, everything is clear to all of you. So, few things I want to give as a summary. It is not for a practical, this is just for your information, for your viva that might be asked with you during the examination hours. What are the factors which influence biodiversity? So there are 2 types of factors. One is ultimate factor. Here, habitat and heterogeneity is the first point. Because if there is only one plant, there is no heterogeneity. If number of plants are there, habitat shows heterogeneity. The disturbance, the biogeographical history, like somewhere if a forest is being formed after glaciation or by grazing, so their biogeographical history is also very important. And next is trophical structure. Trophic means herbivores, carnivores, primary consumer, you are making the pyramid. So that is also very important. And rate of evolution. Everywhere there is evolution. And climate is of course very important factor, and primary productivity beside all the things of anywhere's biodiversity. And another is proximate factor, like area. What type of area is this? Then longitude and latitude, they are very important structure, which is mentioned in terms of latitude and elevation. How much level is elevated from the sea level? They are the factors which influences this. So what conclusion you can draw by studying this biodiversity? So this biodiversity may be influenced by many factors. Interaction among drivers. Who are the drivers? The producer, consumer, if they are interacting, so it results a complex pattern of biodiversity. If only one plant is there, there is no biodiversity. So there will be no interaction. So there will be no complex pattern. Taxonomic group and guild. Guild means the community which is residing there. So taxonomic group, the plants or animal, whatever is living there, is directly or indirectly associated with the society of that particular area. Anthropogenic, you may say. So despite of complexity, major factor can explain up to 90% of the variation in biodiversity is locally to globally. Because from local, that is from alpha, you go to the gamma. From alpha, you can go to the gamma. So local is also important, which plays directly or indirectly role on the global pattern. Thus, global prediction about patterns of biodiversity, driving factors and consequences for managements are possible. So always people think about local area alpha diversity is very important for control of any biodiversity. So I think this is all about your practical with some questions about the different Viva examination. So I think you now you can do excel in your examination because in books so many lengthy process of diversity is there that is very easy to determine within a moment. So I think you have no problem. Okay. See you next time. Thank you very much. Wish you all the best.